Hello friends, welcome to this channel. Today we are going to see about the study of microbial structure. And to study the microbial structure, the first thing is to see the microbial structure. To see the microbial structure, what we need is microscope. So let's begin. What is microscopy and how it works? So before going to the microscopy, we must know about some basic values, some basic mathematical expressions that will be used in microscopy. So the first thing is you must know that 1 centimeter is having 10 to the power minus 2 meter, 1 millimeter is having 10 to the power minus 3 meters, micrometer is having 10 to the power minus 6 meters, nano is having 10 to the power minus 9, and angstrom is 10 to the power minus 10. And out of these, minus 10 means micrometer and nanometer are very frequently used values. And angstrom. These three are very important. Micro means 10 to the power minus 6, nano means 10 to the power minus 9, and angstrom means 10 to the power minus 10. If here is meter, you can put meter here also. Okay. So after this, we must know about the wavelength. This is very important property used in microscope. This is the property of light, and light, light is a wave nature. Light is wavy in nature, so it moves like up, down, up, down, up, down, up, down. So the distance between two upper points or two lower points is known as one wavelength of light. The topmost points are known as crest and the lower points are known as top. So this value is very important in microscope, and this is represented by lambda, that is Greek letter. This is the expression of wavelength. And next thing is resolution. Resolution means this is the ability of any lens of our eye to see two items as separate and discrete unit. That if someone is having eye with better resolution, so 2 points that I can see separately in an easy manner. This can be explained in these two points. These 2 points separate entities, but this can be seen like merged points with a less resolution eye or lens. Okay, so to see 2 points separately, the important condition is that light must pass between 2 points. Light must pass between two objects. This is the distance. Light must be able to cross between these 2 points. Then only you can see these points separately. Okay. So if wavelength of any light is higher, will you able to see these points separately? The answer is definitely no, because light must be with lesser wavelength so that it can pass easily between these two objects or points, and we can see these points separately. Okay, so the key to resolution is to get light of short enough wavelength to fit between the objects that we want to see separately. And the average wavelength of visible light is 550 nanometer, and The resolution, that maximum resolution with this light is 220 nanometer. Okay, and this is the VIBGYOR. You all, I think you all are aware with the VIBGYOR, that is visible light with average wavelength of 0.55 micrometer or 550 nanometer, and this is from violet to red. VIBGYOR. Okay, as the wavelength of light is going to increase, you can see here this is 0.42 micrometer and this is 0.68 micrometer. As the wavelength of light is going to increase, you can see here also the distance between two points are increasing. The wavelength is increasing. So for better resolution, you must use light from this side, means either green, blue or violet. The light with blue is having better resolution. And next is properties of light. You must be well known that how light behaves when it going to interact with any object or when it falls at any object. So whenever light falls at any object, it can reach to that object and come back. This is known as reflection. If the light strikes an object and bounces back, this is known as reflection. Light can pass through the object also without any disturbance. Then it is known as transmission. Light can reach to any object and some of energy may be absorbed by the object, so that wavelength of light changes. So if one object absorbs some light and emits light with different wavelength, this is known as absorption, and the phenomenon this may have fluorescence also. This occurs in fluorescence also. This light can have fluorescence property also. Okay, and when light falls on any object and cross that object with some angle, then it is known as refraction, and this angle is known as angle of refraction. Okay, this is very important figure to know about microscopy. Next thing is, now again come to resolution. As we know that resolution is ability of any lens or our eye to separate or distinguish between small objects that are close together. So one thing that is important for resolution is lambda or wavelength of light. What next is important is the refractive index. Refractive index of lens. It can be lens of our eye or it can be lens of any microscope. So generally the refractive index of a glass lens is 1.5. Okay, and one term is there, numerical aperture of the lens. Numerical aperture means ability of the lens to gather the light. Means if one lens is collecting more light, it is definitely will have better resolution. Okay, so how you can calculate resolution power of any microscope? That the equation is 0.5 lambda by numerical aperture, and numerical aperture is presented by n sine theta, where theta is this angle. You can see here theta is this angle. This is your sample slide with specimen. Light is coming here. Okay, after this light is going to refract and making some angle. So this angle is going to play important role in calculation of resolution power of any lens. If value of n sin theta is higher, the final value of resolution will be lower. Means this lens is having to resolve less distance, means it is a good lens. Okay, so numerical aperture of any lens is represented by Na, that is n sin theta, and these two values are very important for objective questions, many times asked in CSIR-NET exams, ICMR exams, and many objective questions in different competition. That what is the limit of resolution for a light microscope, that is 0.2 micrometer, and limit of resolution of unaided eye is 200 micrometer. Means our unaided eye, that is healthy eye without any glass, can differentiate 2 points that are separated by maximum 200 micrometer. Okay, fix this value in your brain. This is very important. The resolution limit of unaided eye is 200 micrometer, and resolution limit of a lens microscope, a light microscope, is 0.2 micrometer. Okay, I hope you will remember these 2 points for your exams. And next thing is now come to light microscope. This is the typical image of a compound light microscope. Light microscope means any microscope that is going to use light to make the image, then it is light microscope. That is going to use visible light to make specimens observable is the light microscope. What is light microscope? That is going to use visible light to make a specimen observable. And why this is compound? Because this microscope is having more than one lens. Okay, and this is a standard image of microscope, and these are different parts. Important points to be remembered is that this is the lower portion is having, this is basic stand, this is having a light source. The switch must be back to this microscope. This is the light source, and this is a condenser lens. This is condenser lens. Condenser lens means that is going to collect light from this light source. Okay, that is going to converge light beam. So this light beam will pass through specimen. Okay, this is the light source and this is the condenser. This light will reach to this condenser, and this condenser will make it converge to the specimen. This is a stage and this is general. You can see here is a slide with the specimen. So condenser will send the light to slide, and after that the slide, after passing through the specimen that is on the slide, light reaches to objective lens. Objective lens can have different power of lens, like 10x magnification, 40x magnification, 100x magnification, 25x magnification. Okay, so this is the nose piece. This is the nose piece. This that is having all kind of objective lens. After passing through objective lens, light reaches to ocular lens. Okay, so this is ocular means eye. So this is the end close to eye. This is the end close to your eye. This is like nose. So objective lens, three types of lenses are there. One is condenser, the next is objective, and next is ocular. Okay, and apart from that there is a light source, focusing knob. It can be fine knob, coarse knob, fine for very smaller adjustments like smaller movements, and coarse for fast focusing, and stage is to hold the sample. Okay, so this is the general idea about microscope. These are some points. You can make a screenshot of this and write in your exams. That condenser is the it converges the light beam so that it can pass through the specimen. Objective lens magnifies the image before it passes through the body tube to the ocular lens. This is body tube, okay, and ocular lens is further magnifies the image close to eyes. Need sample to be one very important thing, that to see the sample in light microscope, your sample must be stained to provide the contrast between sample and the background. Otherwise, you will not able to differentiate or see the sample properly. So staining can be like gram stain, acid fast stain, that I will come to later in the next lesson chapter. Okay, this is the general idea about compound light microscope, and you can also draw the path of light, how light passes through the compound light microscope, and definitely you will have very good marks in your exam. So let's begin. How to draw this? Very simple method. First, draw a light. Draw an arrow, or you can put a bulb like symbol here. This is the light, and these are three lenses as I told earlier, na? Condenser lens, objective lens, and ocular lens. Condenser lens ka, what is the work of condenser lens? It condenses light for objective lens. So in first step, and this is your sample here, in first step, and this is I. In first step, light is reaching to the condenser lens. Now this condenser lens will is this lens is going to condense light to the specimen here, and after that light is passing through the specimen, okay, and it is going to objective lens. Objective lens is the first lens that is going to magnify the sample, magnify the specimen, and when this light passes through objective lens to next level, that is the ocular lens, again it makes a cross here, and after reaching to ocular lens, second magnification happens here and light reaches to your eye to make the image of the sample. This is very simple diagram, and this can be made in an easy manner in your exams. Okay, so I hope this is helpful for you. Next thing is this is about light microscope. Then I am coming to dark field or dark ground microscope. As the name is suggesting, dark ground microscope means the microscope image of this microscope is having dark background. You can see here also. So why this is helpful? This is helpful to see living unstained cells, है ना? It will going to produce dark background by a very simple way. How? The condenser lens is having a dark field stop. Here it is, dark field stop. Okay, this dark field stop is having a stop here, and light can pass through these spaces. This apparatus, this setup at the condenser lens is going to produce a hollow cone of light on the specimen, so that in such a way that only light that is reflected or refracted by the specimen forms an image. This is important thing, that light is not going to make the image directly. Only reflected or refracted light is going to make the image. Apart from that, the background will remain dark. Okay, that's why it is known as dark ground microscope, and this is helpful to see live organisms, and these are like special shape like Treponema pallidum, that causes syphilis and leptospira. These are very two important examples, many times asked in the exam, especially in objective question, type pattern of exam. This is very important. The dark ground or dark field microscopy is used to see Treponema pallidum, the causative agent of syphilis and leptospira. So please have some focus with these 2 points. And now I am coming to phase contrast microscopy. Okay, phase contrast microscopy is also useful to see unstained live cells, but with bright background. With bright background. And how this is going to produce? This, in earlier phase contrast we use one stopper. Here we are going to use two stopper. One is with objective lens and one is with condenser. Okay, so this is having this phase plate is attached close to the objective lens, and this annular stop with annular ring is close to condenser lens. In dark field microscopy only one stopper was there. Here one stopper is there with the condenser as well as phase plate with the objective lens, and this special setup is going to produce the light that is having differences in refractive indexes of various structures. What is differences in the refractive index of various structures? Suppose like one cell is having a dense nucleus, and that is surrounded by a cytoplasm. So because when light reaches to that, because of their density, nucleus is going to produce light with different wavelength, and cytoplasm is going to produce light with some different wavelength. So refractive index is going to change. This refractive index, light passing through object of diffractive indexes produces phase difference between rays, leading to produce light and dark contrast in the image. See, in the cell, there are different cell organelles. There is cytoplasm. Everything is having different refractive index. So that difference is in refractive index is going to reflect with phase difference. Means dense part will produce the light with different wavelength, and material with less density or different refractive index is going to produce different wavelength. I mean to say that if there is differences in the refractive index of two different organelles, that is going to produce ultimately image with different wavelength on your eye. So in this way, whatever the image, it will have a phase contrast image. Okay, so you just need to learn that is because of refractive index of various structure, a phase contrast image is going to produce. Okay, so this is all about basics of phase contrast microscopy, and this is how the sample can be prepared for light microscopy. Then you can use the wet mount, or you can make the smears and stain them with the simple staining, differential stain like gram stain and acid fast stain, And there are special staining methods like negative staining, flagellar staining, and endospore staining. This all I'm going to cover in next lecture. Okay, so till now we learned about some basics of microscope. I think this is sufficient for your exam. Thank you so much for watching this video. Bye bye. Take care.